The overall goal of this procedure is to maximize the quality and quantity of data extracted from tissue samples.
This method can help answer key questions in the field of translational cancer research, such as which mutations are harbored, which pathways are activated, and which immune cell subtypes infiltrate human tumors. The main advantage of this technique is that a large amount of data can be generated from a single surgical resection of human tissue. One of the most critical steps prior to specimen processing is to ensure sample quality and validate the presence of viable tumor by pathological assessment as described in the protocol text.
All relevant information for the specimen or tissue, such as patient name, number, treatment, and date of surgery, should be made readily accessible in an institutional tissue database. Fresh tissue specimens should always be stored on wet ice and processed as quickly as possible in order to limit changes induced by extraction of the tissue sample from its natural environment. Normal tumor-matched and tumor tissue samples should be processed separately to avoid cross-contamination. Prepare a sterile Petri culture dish, a scalpel, as well as a needle or forceps for tissue processing. In order to limit the time of sample processing, lay out all required items in advance as depicted here. Place the tissue specimen in the open Petri culture dish, flat against the surface. Inspect the sample from all angles to gain a better understanding of its structure and dimensions.